Okay, my next introduction is Dr. Larry Glass. Dr. Larry Glass is coming to us for the first time from Neuren Pharmaceuticals. For those of you who know, there was a press release earlier this year that Neuren Pharmaceuticals is looking to start a clinical trial in Angelman Syndrome as well. Let's see what year he chooses for that. And I won't give away anything. But he has been with Neuren Pharmaceuticals since 2004 as their chief science officer and working in the development of their pipeline, focusing on CNS disorders. He's a biologist and an epidemiologist, and he has more than 35 years of experience in translational research as a scientist. So we welcome Dr. Glass.
Okay. Well, we're new here. In fact, we're so new in the Angelman world that we almost missed the party. But we tend to move fast, and what I want to do today is give you a little bit of information about the company. And it's more background than it is about, what specifically our drugs we think will do in Angelman, because to us, Angelman is one of a range of conditions that are all connected by various things. So Neuren is a New Zealand company, headquartered in Australia. We are focused at this point purely on rare and orphan neurodevelopmental disorders. The company was actually founded to develop two analogs of IGF-1 derivatives. So that's what we do. All of our programs are conducted under FDA auspices, in spite of the fact that we're in Australia. And all our clinical trials, except phase 1, are conducted in the US. The analogs that we're developing, and I'll talk a little bit about it in the next slide, I'll show some pictures. The analogs we're developing are orally available, whereas the natural forms are not. They cross the blood-brain barrier readily, which a lot of peptides don't. They have improved pharmacokinetics, so they have longer, you can go longer in between dosing. Usually for us, it's going to be twice-a-day dosing. And this is important, they don't bind to the IGF-1 receptor, so they don't come with any of the liabilities in terms of having effects on the peripheral, in the peripheral circulation. So the first one is we call trofinetide. That's the generic name for it. We used to call it NNZ-2591. It's an analog of GPE, which is the N-terminal tripeptide of IGF-1. Right now, trofinetide is in phase 3, a pivotal phase 3 study in Rett syndrome, and we've completed the first phase 2 in Fragile X syndrome. All of the trials have been positive to date. We have, for those indications, orphan designation in the US and Europe, fast track designation in the US for both. That program is partnered with Acadia Pharmaceuticals in La Jolla, and they'll be executing the phase 3, which allows us to turn our attention more to what we just call 2591. We haven't come up with a clever name for it yet. 2591 is an analog of what's called cyclic GP, which is actually a second-order metabolite from IGF-1 to GPE to cyclic PG. And we're targeting what more and more are being referred to as synaptopathies. The word doesn't exactly roll off your tongue very easily, but it basically refers to conditions or disorders or diseases in which the synapses are experiencing pathologic change due to some underlying, in this case, primarily genetic changes. Right now, we're including Angelman, Phelan-McDermott syndrome, and Pitt-Hopkins syndrome. We have orphan designation in the US for all three, and like everybody else who's been up here, we're expecting to start trials, phase 2 trials in 2020. So I told you I'd show you a picture. You don't have to memorize this. IGF-1 is in the upper left. That's the full molecule. And where that little purple arrow, I think that's purple, down at the bottom, it says GPE. In the brain, that's cleaved. So the GPE comes off, and that's what that picture on the upper right-hand side is. That molecule was modified just slightly by us, or actually by a medicinal chemistry group at the University of Auckland, where all this came from, by adding a methyl group so that it would be less visible, less sensitive to degradation by proteolytic enzymes, and which gives it the oral bioavailability and also the longer dwell time duration in when it's administered. And then, as I said, GPE is further metabolized to cyclic GP, cycloglypro, down on the left there, and 2591 is a synthetic analog of that, where we stuck an allyl group in it for the same purpose, which is to make it less visible, less susceptible to proteases in circulation. Excuse me. So let's talk just briefly about what IGF-1 does in the brain and what its metabolites do. You've probably all heard of IGF-1. It's actually the effector molecule for growth hormone. It makes growth hormone work. When IGF-1, which is produced predominantly in the liver, in peripheral circulation, binds to the IGF-1 receptor on cells, it triggers a whole range of different activities related to metabolism and cellular signaling. But in the brain, IGF-1 is not actually crossing from the liver into the brain. IGF-1 is produced in the brain by the cells of the brain, neurons in particular, but also neuroglial cells, which are the sort of resident immune cells within the brain. Microglia are particularly important in this. So the IGF-1 that's expressed in the brain binds to the IGF-1 receptor. What doesn't get bound is very rapidly metabolized to the truncated form, which is down there in the sort of the left side of thing. And both IGF-1 and the truncated form bind to the IGF-1 receptor, and they activate critically important cellular signaling pathways that are important and actually essential for neurons to talk to each other and for synapses to develop and mature. IGF-1, as I said before, is actually then metabolized to GPE, which is eventually metabolized to cyclic GP. GPE regulates microglia, whereas IGF-1 binds to and is produced primarily by neurons. GPE and cyclic GP primarily bind to, although we don't know where the binding site is, but they primarily are found in association with glial cells. And there they regulate the role of microglia in pruning dendrites, which is absolutely essential for the development and maintenance and repair and maturation of synapses. They also affect signaling pathways, in fact, the same ones that IGF-1 does, but that's via their action on neurons, which then go on to produce to regulate the signaling pathways. Cyclic GP also has a unique feature, we think it's unique at any rate, in that it regulates the binding of IGF-1 to the IGF-1 receptor, so that it's regulating the availability of IGF-1 to do its thing in the brain. So talk a little bit about why we're targeting synaptopathies. Neurodevelopmental disorders result from all different kinds of mutations and deletions, but all of them feature deficits in synaptic plasticity. Oftentimes, regulating the strength between the connections between neurons is impaired, and very often the dendrites that form the synapses are too long, too short, too immature, etc. And these problems in signaling and the problems in synaptic morphology can directly cause the behavioral and cognitive and motor and even the autonomic symptoms associated with some of these synaptopathies. So what 2591 does is that it first and foremost reduces the expression of inflammatory cytokines, which neuroinflammation is an important feature of many of these disorders. It also, and this is probably the most important function in the synaptopathies, is that it normalizes microglia. I talked before about microglia being critical for the pruning. I forget who it was, but Virginia Hughes first referred to microglia as the constant gardener. And that's what they do. They prune weak synapses and allow the strong synapses to grow. And without that selective pruning, you either end up with too many or too few, and it varies from one to the next. It also, primarily because of its effect on the binding of IGF-1 to IGF binding protein 3, controls or regulates the bioavailability of IGF-1, so that if IGF-1 is depressed, as it is in many of the neurodevelopmental disorders, then it regulates that and the level of IGF-1 come up. So that it can fix both hyperconnectivity and hypoconnectivity, depending on which is needed. So it really is a homeostatic mechanism. The 251 is an analog of a homeostasis seeking molecule. And because of the role that it evolved with in terms of supporting homeostasis, its effects are state dependent. In wild type animals, it has no effect. I mean, virtually no effect. It's only where there's a need for it. I shouldn't use that word, but I can't think of a better one. Where it's needed, it's active, but where it's not needed, it isn't active. And that creates a pretty good basis for safety and tolerability. And we found that the parent drug, the parent compound, and 2591 so far have a very good safety profile. So unfortunately, I don't have this information from the Angelman model, so I'm showing you something from the SHANK3 model, which is from the Phelan-McDermid world. But the things that I'm showing here are directly relevant to some of the underlying pathology associated with Angelman syndrome. In the upper left there, you see this is the length of dendrites. In the SHANK3 model, in Phelan-McDermid syndrome, the dendrites are actually longer. They're not being pruned effectively by the microglia. And with 2591 on board, this is in vitro, the length of the dendrites actually is returned to normal, no different than wild type, with no effect on the normal. You can see the picture on the upper right. That's green fluorescent staining of the dendrites, showing the reduction in the length of them. In the SHANK3 model, IGF-1 is also underexpressed or underavailable, and that shows on the bottom right there. And the 2591 effectively normalizes that, so that IGF-1 bioavailability is no different than wild type. And the ERK signaling, which is part of the neuronal signaling cascade, is returned to normal as well with 2591 in that model. These are the results on the behavioral side in the Angelman model, going from activity and locomotion on the left upper to then species typical behavior for the next two on the top, which we tend to think of as somewhat related to activities of daily living. You'll see their complete restoration of normal function in the model. On the bottom, we have sociability, which is again completely normalized, as are in the middle physical capacity. This is in a Fort swim test, and a measure of memory and cognition in a modified water maze. So everything with 2591, everything was normalized. You could not really tell the difference between wild type and the knockout mice. It also eliminated audiogenic seizures completely. So I know I'm running over time here. So where are we? We've demonstrated efficacy in a bunch of different models, and we'll keep doing more. And I've already mentioned that we have orphan designation. The 2591 is 100% orally available. So what we're talking about for a dosage form here is a flavored liquid, probably an ounce or less, that's administered probably twice a day. In Rett syndrome, it's strawberry flavored because that appeals to that population and it covers up the native taste of the stuff. I don't know yet what it'll be for Angelman. We're using our experience in developing rapidly. We're too small to be patient, so we've learned how to move quickly and effectively in this strange world we all inhabit. And we're using that expertise to move ahead in Angelman. I mean, trying to talk about phase 2 trials in 2020 from where we are right now is pretty ambitious, but we're pretty confident that we can get there. And we're also going to go straight into a pediatric study. In Rett syndrome, we tested first in adolescents and adults, and then we went into kids. In this case, we're going straight into a 12-week study in a pediatric population. And the purpose of that study, that first phase 2 study, is to try and get a sense of the magnitude of the response, the validity of our outcome measures, and we're pretty confident that we'll be using what we call an anchored CGI, a clinical global impression of improvement, which is anchored to specific symptoms that are relevant to each of these indications. Excuse me. We will incorporate other potential endpoints as we find ones that seem reasonable and predictive. And of course, that phase 2 trial will also confirm the optimal dose for our pivotal trial, which hopefully will be the next one. So that's a summary of where we are, what we do, where we're going. Thank you.
Hi, it says pediatric patients. Can you speak to the age group?
Yeah. Yeah, we don't know. We haven't defined the exact age range yet, but it will be young to teenage somewhere.
Do you know if this will be an open label trial or placebo-controlled is first question. And then the second question for the previous syndromes, the Rett and Fragile X, have those been published studies or do you have results that can be seen?
Yeah. We're not 100% sure whether this is going to be open label or placebo-controlled or somewhere in between. There are variations in between there. We'll be settling that as we move forward. Right now, we're concentrating as we begin to do the clinical planning. We're also doing the really exciting stuff like manufacturing the drug and doing the chronic toxicology studies that have to be done to get FDA approval. So I'm not trying to dodge the question. I don't know yet whether the same as I don't know the exact age range. I don't know the trial design. I don't even know the final selection of endpoints. But that will be forthcoming over the next months.
And then
there was a paper in Neurology about, Liz, what was it, about 3 months ago, something like that? About 3 months ago on what we call Rett 002, which was the second Rett study in pediatric patients. The Fragile X study has been presented, but the manuscript has not yet been published.
Thank you.
I have a question over here. Hi, me. I have a question.
Oh, hi.
So last year we heard a wonderful presentation about the benefit and the phenotypic rescue in the mouse model with IGF-2. And I'm wondering if you can give us a little understanding briefly about the differences between IGF-1 potential effect as a synaptopathy the benefit to a synaptopathy as compared to IGF-2? You have 30 seconds or less. No, I'm just kidding.
30 seconds, yeah. IGF-2 tends to be more active within the CNS and to be more specific to PI3 and AKT-mTOR. Beyond that, I really can't say a whole lot about it. They're both involved in synaptopathies, either increase or decrease of bioavailability and/or expression. It's hard to tell which is which, but it's not clear whether those are whether they're genuinely different effects or whether it's an IGF family that's doing it. I mean, the IGF family, the whole IGF pathway is phenomenally complex. I mean, we talk about with cyclic GP and 2591 regulating the bioavailability of IGF-1 is part of the autoregulatory process that drives that IGF-1 pathway. But there's a lot about that pathway we still don't know. That was a really vague answer, wasn't it?
If you want me to summarize that, the answer is I don't know. Just kidding. Is there another question? I see a hand.
Is NNZ, can you build up a tolerance to this, or is it not likely to build up tolerance?
We don't think there's any, I mean, given the pharmacokinetics, it's unlikely. But to be clear, and I didn't say this and I should have, is we're not talking about the same thing that you're potentially looking at with gene therapy, where whether it's a cure or whether it's a dramatic change in the function. We're talking about something that's, you get up and you take with your cornflakes and you take it before dinner and it has a pretty dramatic effect, but it's something that's likely to have to be taken for life. We don't think this is not going to cure anything. We're not changing the mutation. We're not changing the availability of the proteins. We're sort of running around doing an end run around the downstream consequences of the fact that the protein is not available or is not as available as it should be. Are we going to do the